The title of my talk is No Filter, Sensory Processing in Autism. But before I start to talk about autism and sensory processing and sensory filtering in autism, I want to quickly tell the ones that don't know where the University of Western Ontario is located, show where I actually am. So this is the blue area here is Canada. I tried to put a dot where I think Memphis is. The University of Western Ontario is not in the west of Canada and also not in the west of Ontario, so I have no idea why it's called that. We're actually located on the southernmost appendix of Canada, here. If we zoom in, so this is where London is located. We have a Thames River and Piccadilly Road and everything that the other London has as well. And we are in the middle between Detroit, Toronto, and the Niagara Falls, and nestled between all these big lakes. So we have a lot of snow in the winter, but at the moment it looks more like here. So we have a nice Indian summer. So with that out of the way, let's first start with autism. I'm aware that you had Chris Rowan giving a talk in September who talked a little bit about autism already. So very, very briefly only, autism spectrum disorder, that's how it's called today, is an umbrella term for a number of overlapping disorders, including autistic disorder, Asperger's, Rats syndrome, and other, again, overlapping disorders. Nowadays, 1 in 68 children in North America are diagnosed with autism spectrum disorder. And this is, the incidence is increasing. It's not clear whether there are really more children with autism nowadays or whether they are just diagnosed more often. In any case, it's clearly a neurodevelopmental disorder. Kids can often be diagnosed really early. And I think we all think that children are probably born with autism, and the impact occurs during the development, probably the prenatal development. As for the cause of autism, there are genetic and environmental causes that have been associated with autism. Only a very, very small percentage of autism cases are linked to a clear mutation in one specific gene. And these are the syndromic cases like Rett syndrome, where we know a gene, where we know the mutation, and it causes a disorder that is within the autism spectrum, or that has at least core symptoms of autism. But this is a very small percentage. Most cases are a combination of maybe genetic and environmental risk factors with more genetic or more environmental. Like many other psychiatric disorders, we don't often really know what is the cause. In order to diagnose autism, people use the Diagnostic and Statistical Manual of Mental Disorders, the DSM-5. And that means autism is an intrinsically human disease, right? Because DSM-5 outlines 2 clusters of symptoms that are very human symptoms. So one cluster are the persistent deficits in social communication and social interaction. And the other cluster are restricted and repetitive behavior. And within that cluster, we find not only the stereotype behavior and then the insistence of sameness, highly fixated and restricted interests. We also find the hypo or hyperreactivity to sensory input, an unusual interest in sensory aspects of the environment. And this is what we decided to focus on, these hyper and hyporeactivity to sensory input. We think that these sensory aspects of autism and other psychiatric disorders actually provide a window of opportunity for translational studies using animal models. And I think we all agree we need to use animal models to actually figure out what goes on in the brains, what is wrong, what cellular, molecular mechanisms are different, and how can we maybe treat these children or these people. So the sensory symptoms of autism, again, they have become a core symptom of autism with the DSM-5. What is great about the sensory pathways is that they are relatively well described and relatively easily accessible. So in contrast to, for instance, if you think of the social aspects, the social symptoms of autism, the networks responsible for social behavior are very complex. They include huge cortical areas. We don't understand them very well. So it's hard to study them, but it's also, of course, questionable to what extent these complex networks are present in an animal. So the translatability there is not as great as it is in the sensory pathways that are very, very highly conserved between species. And again, very well described. If you think about these sensory symptoms, they actually have, people have shown that they predict the severity of other symptoms. So if the sensory symptoms are really strong, other symptoms, the social symptoms, the stereotypies and other symptoms are often also very much more severe than as predicted by sensory symptoms. So this means that probably there's a common underlying mechanism. So if there is a gene defect or an environmental impact, that could also be epigenetic or could, you know, be whatever type of impact, that it probably impacts the sensory pathways the same way as other networks in the brain. So if we look at what goes wrong in the sensory pathways, we learn of what might be wrong in other pathways as well. It is also debated to what extent the sensory symptoms actually are a cause of at least some of the other symptoms. Because if you think about that sensory pathways are impacted and someone perceives the world differently from other people, it is very difficult, or it can be very challenging to function in a social context if you realize that you are different and differently perceiving things than other people. And so some of the symptoms can actually be directly related to the perturbations in sensory processing. To what extent, people are arguing and debating. So taken together, we think that studying sensory autism symptoms provide a great opportunity for studying disease mechanisms using an animal model, and also to test potential therapies in the animal model. Now, having said this, I just told you in the slide before that autism is an intrinsically human disorder. And so the question is, of course, what type of animal model can we use to study autism? And I want to say it very clearly, there are no autistic animals. Again, it's an intrinsically human disorder. But what we can do is we can look for animal models that model a group of symptoms that we see in autism, and then focus on these group of symptoms rather than calling it an autistic animal. And so this is what we do. We look for animals that have a high construct, phase, and predictive validity. This is the gold standard, I think, for animal models. Construct validity means that there is, for instance, a gene that causes the disease that we then also modulate in the animal. Or if there's an environmental impact, that we model the same environmental impact in the animal. So it's constructed the same way as the disease in humans. The phase validity means that the phenotype is presenting similarly than in humans. And the predictive validity, and this is where a lot of animal models then fall short, is that if we treat them or if we make a prediction based on the animal model on the human situation, that it then holds true. And unfortunately, this is often not the case. So in terms of construct, there are different environmental and genetic factors that we know increase the risk of autism. I listed 3 environmental aspects here. One is prenatal exposure to valproic acid, which is an anti-seizure medication. It is very inexpensive and it's very effective. However, it increases the risk, if taken during pregnancy, it increases the risk for the child several folds to actually have autism. To have a seizure during pregnancy, although is also very dangerous. So there are women that are pregnant that do take valproic acid. There's also maternal immune activation during pregnancy. So it has been known that strong infections, viral or bacterial, during pregnancy impact the fetal brain development. And this is modeled in animals by injecting poly-IC, which is a synthetic double-stranded RNA-like molecule that the immune system thinks it's a virus, and so it mounts an immune response. Or lipopolysaccharides, which again is something that normally is in the walls of bacteria, and the immune system then ramps up an immune response. And it is the maternal immune response that actually impacts the fetal brain development. Another environmental factor is old age of the father, and so on. There are an entire list of other environmental risk factors. In terms of genetic risk factors, again, we have the syndromic cases, like there's a Fragile X syndrome, there's Rett syndrome, and one single gene genetic cause for an autism-like syndrome is the Cntnap2 rat. So this is the rat that we use in my lab, along with poly-IC and valproic acid models. The Cntnap2 gene codes for a protein called Caspr2. It's a contactine-associated protein. It is heavily expressed during development, and it's mostly expressed specifically in sensory pathways, which of course makes this a good model for us, and in cortical areas that relate to sensory processing and to language. It is a rare single gene cause for a severe form of neurodevelopmental disorder with core symptoms of autism. And the strongest evidence comes from a population of old order Amish people in Pennsylvania, where 13 individuals have a mutation in Cntnap2 that introduces a stop codon in the gene. And so their Cntnap2, their Caspr2 protein is rendered completely dysfunctional. And these 13 individuals have the severe form of neurodevelopmental disorder, but they show core symptoms of autism. They are nonverbal, so they can't speak. They learn to speak a little bit and then they regress. They also have seizures and intellectual disability to a different degree. Some are rather severe and some not so severe. It can be caused also by seizures. So the intellectual disability is not a strong symptoms in these individuals. Apart from the strong evidence in this population, a lot of mutations and genetic variants, SNPs, have been associated with autistic spectrum disorders by a multitude of studies. And just a little bit about the Cntnap gene. The Cntnap gene is the biggest gene in the human genome. It's located on chromosome 7. It contains 24 exons. It's, I think, 2.3 megabase big. It has several transcription factor binding sites. One is a binding site for FOXP2. And if you ask any neuroscientists about FOXP2, they immediately associate that with language. So Cntnap2 is regulated by FOXP2, which might be the reason why these individuals with a Cntnap2 loss are nonverbal, so they don't learn. Songbirds with a lack of catnap, where we inactivate Cntnap2 genes, they also do not learn their species-specific song. So Cntnap2 has a linkage to language, not only in humans, also in animals. There are a multitude of mutations. There's all these different colored arrows and bars here, and they are linked to different psychiatric disorders, all of them with great comorbidity to autism. There is the Caspr2 protein is a protein with a large extracellular domain, cellular part with a lot of different binding domains. It has a transmembrane part, just one, and then an intracellular domain with a PDZ domain. It works as a cell adhesion molecule. So it is a family member of the large family of neurexins. And as this cell adhesion molecule, it mediates interaction between neurons and glia during development. So it is crucial for myelination of axons. It also clusters potassium channels in differentiating axons, which are important for the conduction of neuronal signals. And it also stabilizes synapses between different cell types. So in general, we can say Caspr2 is important for the conduction of signals on neural pathways, but also for stabilizing synapses, specifically inhibitory synapses on excitatory neurons and vice versa. And so I know that Chris Rowan spoke a little bit about this disequilibrium between inhibition and excitation in autism, which also is one reason why there's a high comorbidity with epilepsy, because there is a lack of inhibition. And so Cntnap fails to stabilize GABAergic synapses on glutamatergic neurons and thereby less inhibition. Importantly, Caspr2 and the Cntnap2 gene are highly homologous with, between rodents, mammals, and humans. Between mouse and human, there's 94% of homology. And between chimpanzees and humans, 99.5% homology. So highly conserved. So again, we use this Cntnap2 rat. Most importantly, the Cntnap2 knockout mice and rats have been created, and they show core symptoms of autism. They show stereotypic behavior. They show changed sociability, and so on. But what we are focusing on, and I'm focusing on in this talk, is sensory processing. So what does sensory processing look like in this rat? And we focus on auditory processing, which is, again, mostly language related, but it's also easy to control. We can play stimuli, different auditory stimuli, loud, fast, slow, or quiet. We don't have to work in the dark as if what we would have to do if we used visual stimuli. So auditory stimuli are just great to work with. And also we have a lot of different readouts of auditory processing at different levels in the auditory processing pathway. So this here is a figure of the ascending auditory pathway. It starts the hair cells in the cochlea in the ear that are innervated by the spiral ganglion cell, which makes synapses here in the cochlear nucleus. And then there are synapses here in what we call the auditory brainstem nuclei. So this is a little bit of a simplified pathway. And from there, the neuronal signals ascend through fiber tracts that are called the lateral lemniscus to the midbrain. There's a relay station here in the superior and more importantly, inferior colliculi. And from there, it goes through the thalamus and finally to the auditory cortex. So how do we measure auditory activity? I'll start at the bottom. So we can measure auditory brainstem responses. These are electrical responses that we can measure in humans by just gluing an electrode here on the bone behind the ear. And when we play a sound, we have these very characteristic waveforms here. And each peak in this waveform actually mirrors the neural signal at different stations along the ascending auditory pathway. And I have coded them here, referring to the peaks here in the auditory brainstem response. So it is a measure that we can do in humans and in animals alike, and it gives us information about neuronal signals in the brainstem here. The next thing we can do is we can measure acoustic startle responses. That is a reflector response, muscle twitch, when we startle in response to a sudden acoustic loud noise. And we know the acoustic startle pathway. It's a very short pathway because it's a very fast reflex. It includes the spiral ganglion cell, the cochlear nucleus here, and from there, neurons go into the reticular formation, which is this region here. They innervate these PNC giant neurons, the startle neurons in the reticular formation that then directly innervate motor neurons and that make our muscles twitch. So the acoustic startle response is a readout of very early auditory processing here. It is a behavioral response to auditory stimuli on the very basic brainstem level. We also can measure prepulse inhibition of startle, which is the inhibition of a startle response, and I will explain that in more detail a little later. But it's the inhibition of the startle response by a prepulse, by another auditory or visual or tactile stimulus. And we know that there's a feed-forward inhibition through a very short pathway again that includes some midbrain structures, namely the colliculi here, and a structure called the pedunculopontine tegmentum that then inhibits the startle pathway. So by measuring prepulse inhibition of startle, we have a measure of the function of these midbrain nuclei here in the brainstem because they are mediating prepulse inhibition. And then we have other readouts like electroencephalogram or acoustically evoked potentials, where we, of course, measure cortical responses to a sound. And we can perform auditory perceptual tasks. So all these tasks, prepulse inhibition and acoustic startle responses, they are reflectory responses. They're implicit. It doesn't need any motivation or any voluntary behavior of the animal or a human. Whereas the auditory perceptual task, this is where we ask a human or a rat, is this loud? Is this quiet? Is this fast? Is this slow? And so on and so forth. So this really probes for the cortical level of information processing and for perceptual tasks. So in this talk, I want to focus on the brainstem tasks. I will not talk about the perceptual tasks. I will only rather quickly talk about these readouts here of the brainstem. And then in the second part of my talk, I want to then drill more into the mechanisms that underlie these measures and how they might be perturbed in animal models for autism. But we can talk about these other tasks in the discussion if there's interest. Okay, let's start with the auditory brainstem response. As I mentioned, a very characteristic waveform, and we can, of course, measure the amplitude of the different peaks, and we can measure the latency of different peaks. And it's very easily measured in children with autism of all ages. We just glue an electrode behind the ear, and these are the reference electrodes here on the forehead, and then play sounds and measure the response to that. We do that in the same way in the rat. So we actually introduce electrodes underneath the skin in the anesthetized rat, because also an anesthetized rat still hears, and put it in a soundproof chamber and measure the auditory brainstem response. So what we know in kids with autism, and this is not our data, this is data from others, is that auditory brainstem responses, that the amplitude of the waves increase during development, and the latency of these peaks in the wave decrease during development. And we see this in the study here. So we have the left part of this figure are newborn infants between 0 and 3 months of age. And we see the kids that are going to be diagnosed with autism later on, and control typically developing babies. And we can see that even shortly after birth, in this case, this is the latency. The latency of the auditory brainstem is already increased compared to typical developing kids. As these kids mature, and these are kids now and 1.5 to 3.5 years of age, we can see that the latency decreased, so the auditory brainstem responses got faster, but much more so in the typically developing kids than in the children with autism. And on the right side, we see a meta-study. Each blue dot would be a single study, a paper, where they looked at the same latencies, and we can see that, and I apologize, you can't read the x-axis, but on the x-axis, this is 0, so this is our newborn children, And we have 18 years of age here and 9 years of age here. And we can see that across all these studies that the latency of the auditory brainstem response normalizes when the children reach adulthood. So the green line, that's what it would be the normalized latency in typically developing kids, and the red line is the latency in children with autism. So children with autism show a delayed brainstem maturation. It looks like they just lag behind. The waves are smaller, but also slower, and eventually it catches up when they become adults. So we looked at this in the Cntnap2 rat. And what we see is exactly the same. To an extent, we were almost astonished. So we look at postnatal day 28, that's shortly after weaning. These are fairly young rats. And if we measure, in this case, we looked at I think these are the latencies as well. We can see that the latencies of wave 1 is slightly increased, wave 2 even more so in 3 and 4. However, when the same animals actually age, this is P42, this would be an adolescent, and then P70, which would be a young adult, so maybe 18 years of age. And we see that these latencies normalize. So like in kids with autism, we seem to have this delayed maturation of the auditory brainstem. And this is true for the latency, but also for the amplitudes, which it's in the paper, but I don't show this here. We also have some other measures where we measure the interpeak latencies, so between peak 1 and 2 and between peak 2 and 4, and we see the same trend. So the latencies get shorter across development, but also the differences between wild type and knockout. So wild type is always blue here and knockout is always red. We see that the knockouts have longer latencies, but eventually they seem to catch up with the wild type animals. So this is auditory brainstem recording. Let's move on to the next level up, which is the behavioral outcome, the acoustic startle response. And again, we have a nice translational measure because we can measure the muscle twitch in humans by actually measuring one component of the startle response, which is the eye blink. When we startle, we close our eyes. And so we measured an electromyogram of the muscle just below the eye that makes the eye close. In humans, in animals, we do that a little easier, simpler. We just measure the entire whole body muscle twitch by putting the animal on a motion sensitive platform. And then have a transducer underneath that converts the movement of the platform into a voltage. What we know about acoustic startle response in children with autism is that they are slower, which is not surprising. The auditory brainstem is slower, so the startle is slower as well. But the amplitude is also increased. So this is a study by someone else, Hiroshi Takahashi, that showed that the latency is longer, and this is data from our own lab showing startle to different loudness, different volume startle stimuli. So in children with autism, so children normally start startling at around 85 decibels, loud sudden noise. But we can see the blue line is kids with autism. The other are different types of control, typically developing children. And we see this left shift in the I/O function. So children with autism startle at a lower noise level already. So they have a higher but a slower startle response, both children with autism. And we looked at this in the Cntnap rat, and we see a very similar situation. We look in the young and in adults, and we see a slight increase of startle, especially at these quieter startle stimuli in the young ones. Top is male, bottom are females. But this is even more pronounced in the adults. So as opposed to the delayed maturation in the brainstem, when we look at the behavioral outcome, the behavioral readout, we actually see a persistent increase in startle response that persists in adulthood. And we see that even more so in males than in females, which is also something that is true for humans with autism. So CatNap knockout rats show this left shift of the startle eye function and much greater reactivity, especially at lower intensities and also in the adult. I also want to mention one startle modulation, which is habituation. So if we repeatedly startle someone, normally we would habituate to the startle response. Kids do not habituate that well, but we see in different control groups here, at least, you know, they go slightly down, slowly down a little bit. What we see in children with autism is a huge sensitization. At least an initial huge sensitization in this study. So children with autism do also not habituate as well to repeated startle stimuli as other children. And this maybe some people can relate to these anecdotal stories about kids with autism that get easily distracted and cannot tune out background noise or repetitive noise made by other children or by other things. They actually sensitize rather than habituate to these type of stimuli. So habituation is one way to measure this. And we find, again, exactly the same phenotype in our CatNap knockout rats, that they show strong sensitization instead of habituation, especially in the young animals. They do the adult animals then do habituate. So they seem to, at one point then, I don't know, learn to habituate. The other modulation that we show is prepulse inhibition. So prepulse inhibition, I mentioned that before, that is the reduction of these acoustic startle response through that midbrain feed-forward inhibitory pathway. And it occurs when there is any type of prepulse, visual, tactile, auditory prepulse that does not startle the animal. But what people think it is, it elicits an orienting response to that prepulse. And this inhibits the startle response, so the motor response. And prepulse inhibition has been actually originally developed in people with schizophrenia to measure this lack of what we call sensory motor gating of sensory stimuli. And there are a lot of studies of people with autism on prepulse inhibition. Some show a lack of prepulse inhibition, some show increased prepulse inhibition. So that really seems to depend on the type of autism within the spectrum, whether or not an individual with autism shows impairment of prepulse inhibition. Our rats, our CatNap rats clearly show an impairment of prepulse inhibition. On the top here, we see the baseline startle. So as shown before, they startle much higher, but also they have less prepulse inhibition. So that is the percent inhibition, the percent reduction that we see by a prepulse. And this is the percent reduction in wild type, in young animals, and then in the CatNap young animals. And these are the same animals in adult. And again, we see this disruption, the lower amount of inhibition in the CatNap animals, which is more pronounced in the adult again than in young animals. So the adult is the right one. The young ones are here on the left. And these are just different prepulse stimuli with different interstimulus intervals, 30 milliseconds and 100 milliseconds given before the startle pulse. Prepulse inhibition, again, is an implicit reaction. It has nothing to do with learning, and it occurs subconscious. We cannot influence our prepulse inhibition. And just as a reminder, it is mediated through this fast feed-forward inhibitory circle. Now, so these are the behavioral things that we measure in our CatNap rats on the brainstem level. I want to just do an interim summary. So what I have shown you so far is there is a delayed maturation of auditory processing in the brainstem. They startle higher, but there are also impairments of habituation and prepulse inhibition. And that points out that there are actually sensory filtering impairments. So both habituation and prepulse inhibition are measures of sensory filtering, and they actually parallel findings in humans. And I want to switch gear a little bit and spend only like 2-3 minutes to talk on the importance of sensory filtering, because I think many of you might actually not be really aware what sensory filtering is and how important it is. So, and my lab actually studies mechanism of sensory filtering. This is my core business, so I can't help but talk a little bit about sensory filtering. So to the students, the big question is, what is the input into our brain? Like anything that goes into our brain. The answer is it's sensory information. Everything that goes into our brain comes from our senses or from some sensors inside our body that senses all kinds of things like our blood pressure and salinity and osmolarity and so on, which we will never become aware of. Nevertheless, all these signals are constantly reaching our brain. And this is anything that goes into our brain is sensory information. That is the sole input. Then our brain has to process and store it and modulate and modify it. And what it actually does is it comes up with an output. And the output of our brain is some form of behavior, hopefully appropriate behavior, whether this is drinking or, you know, socializing or startling or whatever. So the problem is that we cannot switch off any of our senses. We can close the eyes maybe, but anything else, the senses continuously provide input into our brain. And so there are mechanisms that reduce the load of sensory information constantly, non-stop bombarding the brain so that the brain can actually focus on the stuff that is important and salient. And there are different levels where the sensory information is reduced. It is on the receptor level. I think most of you know that sensory receptors and have experienced that sensory receptors adapt to constant stimulation. So there we have a reduction of information. On the very other spectrum, we can voluntarily focus our attention on something and try to ignore the rest, and thereby reducing, hopefully, the load on our cognitive function. But a lot of the sensory information is actually filtered along the sensory information processing pathway. So these are built-in neuronal mechanisms, such as the mechanisms that lead to habituation and prepulse inhibition that filter or gate sensory information. And these sensory filtering mechanisms are pre-attentive. So they are before the information actually reaches our cortices and our conscious perception. So we are not aware of these, which also means that unfortunately we tend to ignore them. So how important sensory filtering is, as opposed to be constantly bombarded by sensory information, is very difficult to demonstrate to you because your brain is filtering out the information constantly. The closest I can get to that is by showing this nice little painting, which is from Peter Bruegel the Elder. It was painted in 1562. And the name is The Fall of the Rebel Angel. Now you have looked at that picture for quite a while I explained what kind of picture that is. But if I would ask you what is on this picture, what does it actually show? What is the main message? You would have troubles. Why? Because it's so cluttered. There's so much stuff going on that it is really hard to get the main message. And this is what it is like probably for someone who has sensory filtering problems. There's so much stuff reaching the brain that it is hard to get the main message, to actually figure out what's going on. And I bet, I mean, you can look up other paintings of Peter Bruegel. They all look like this. So I'm pretty sure that nowadays he would actually be diagnosed with being somewhere on the spectrum. Okay, so, whoops, hang on. No, oh yeah, okay. So again, I want to switch gear now, having hopefully convinced you how important sensory filtering is for our functioning in our environment. I want to spend the last maybe 10 minutes or so, maybe a little less, on looking at cellular mechanisms. So now we have these disruptions in habituation and prepulse inhibition, in startling per se, in the auditory brainstem response. So how can we now leverage the animal model to actually look at cellular mechanisms underlying these disruptions? And of course, one arm of my research, which I'm not going to talk about, is this habituation of the startle response. So I have been working on habituation of startle responses for almost 20 years. And we know that this is the startle pathway, again, a very short pathway. And we know that habituation of startle now occurs through synaptic plasticity here at this synapse of the cochlear neurons projecting to the PNC giant neurons in the reticular formation. A postdoc of mine several years ago has shown that this depends on big potassium channels, which are exactly these type of potassium channels that are clustered by Cntnap2. So I have another postdoc in my lab, Yuri Kahanovic, that currently does patch clamp recordings in the slices, brain slices of rats. And he looks at synaptic plasticity here at these neurons innervating the startle neurons in the reticular formation, and looks whether there's a difference in these BK channel function and in the catnap knockout rat that might actually underlie their impairment in habituation, and whether we can target, for instance, BK channels or other targets in order to improve habituation and change startle reflex in these kids. Again, always assuming that if we can counteract these impairments in startle, maybe we counteract other impairments that are dependent on these BK channels in other brain areas as well. So this is just one arm. I'm not going into much detail there. I want to show a little bit more about the work we do into prepulse inhibition. Again, prepulse inhibition is mediated by this feed-forward midbrain loop that then inhibits PNC giant neurons. And for the better of, I would say, 30 or 40 years, it was always like known that this is a cholinergic projection that actually inhibits the startle pathway and that mediates prepulse inhibition, so that when the prepulse activates these cholinergic neurons in the pedunculopontine tegmentum that then inhibit the startle pathway. And the reason people think it's a cholinergic function is that the PPT, the pedunculopontine tegmental nucleus, I will call it PPT from now on, is one of the very few cholinergic structures in the mammalian brain. And so when they lesion the PPT, there's no more prepulse inhibition, which is weird because, again, schizophrenia is associated with a lack of prepulse inhibition, so is autism. There's no indication that, that acetylcholine plays any role in schizophrenia or autism. But nevertheless, that was a long-standing dogma. So what we did is we used neat new techniques, optogenetics, in order to test this hypothesis. So what we did is we injected an optogenetic virus here into the PPT. So this is the PPT, the pedunculopontine tegmentum. And this is the projection to the reticular formation here where it inhibits startle. So Erin Azopardi, a PhD student in my lab, and Caro Wagner, who was a German exchange student, they used Long Evans transgenic rats that would express Cre recombinase under the chat promoter. That means only cholinergic neurons would express Cre recombinase. Then they injected a virus into the PPT of these rats. And these are, so it's an adeno-associated virus that has a promoter, a very ubiquitous promoter. So all neurons express this promoter, elongation factor 1. It also contains a channel rhodopsin, which is a light-sensitive ion channel that depolarizes the neuron when activated by light. It is tagged with a yellow fluorescent protein so that we can see which neurons are actually expressing our construct. But it also is behind a or after a double inverted open reading frame, which means in order for this to be expressed, it needs Cre recombinase. So that means only cholinergic neurons will actually oppress the virus, because only they express Cre recombinase. And we have a control virus, of course, which has everything but the light-sensitive ion channel. So this viral construct is now injected stereotaxically into the PPT. In the same surgery, we implant a light rod, a light fiber into the PPT. Then we wait for 2 weeks for the virus to become expressed by all cholinergic neurons. And then we can activate these neurons by light. So we tether the animals to a laser, and then we can activate for milliseconds or seconds, for very short intervals, we can activate this light fiber that should activate, depolarize all these cholinergic neurons now that express our construct. So according to the theory, what we think should happen is, if this cholinergic projection here mediates prepulse inhibition, we should be able to inhibit startle by activating these neurons. So not just only by a short acoustic prepulse. Instead of an acoustic prepulse, we should be able to activate these neurons by light, and we should have the same effect, an inhibition of startle. So long story short, we activate, and we use different interstimulus intervals between the activation and the startle pulse. So on the left side, we now see the animals with the acoustic prepulse. We have to show that, yes, they show prepulse inhibition. So the acoustic prepulse of all types of animals was between 80, well, let's say between 50 and 95% of inhibition of startle by an acoustic prepulse. All the blue ones got the real construct, all the gray ones got the control constructs, and these are just control animals for now different types of experiments where we use different frequency of light stimulation in order to activate these PPT neurons. So what happens if we activate these PPT neurons, not by sound, but with a light that should depolarize these neurons? The answer is nothing. So we activate the cholinergic neurons. They probably all fire action potentials, but it doesn't influence startle. Actually, when we activate them with the highest frequency, the highest intensity, the startle is actually higher. So there's no, this is again the amount of inhibition. There is prepulse facilitation. The animals startle higher. And of course, this was accompanied by a huge amount of control experiments. We did in vivo electrophysiology, patch clamp recording to show that these cholinergic neurons really get activated and fire action potentials when we stimulate them by light. We did expression studies showing that more than 90% of the cholinergic neurons really did express the construct. We did even a positive behavioral control showing when we activate these neurons, we can induce condition-place preference, which is another function of these cholinergic neurons here in the PPT. So it looks like it's not the cholinergic neurons here that actually mediate prepulse inhibition. So what else is it then? And the next step that we did is we took a step back and we said, okay, in the past, people have done lesion studies. They lesion the entire PPT, but the PPT contains 1/3 cholinergic, 1/3 GABAergic, and 1/3 glutamatergic neurons. But maybe our technique is also not good enough to actually induce disruption or prepulse inhibition. So what we did in our next experiment, well, what we did in the next experiment first is we inhibited the cholinergic neurons using DREADDs, which is another genetic model. DREADD stands for designer receptor exclusively activated by a designer drug. And so these, again, constructs are injected into the Long Evans Cntnap2 rats. So only cholinergic neurons express the constructs. But the constructs in this time, they contained an artificial receptor that inhibits neurons. And it gets activated. Hang on, so we inject that, we let the cells express this, and then we can activate the receptor by the designer drug, which is clozapine N oxide, which presumably does nothing to the animal, but it will inhibit specifically these neurons that express, these cholinergic neurons in the PPT that express our construct. And again, long story short, when we do that, and we can now compare within the animal, within subject, before and after injection of designer drug, we don't see any effect on prepulse inhibition. Prepulse inhibition works just fine. We can inhibit specifically the cholinergic neurons in the PPT. It still works. They do show lower startle responses in general, though. That means that cholinergic neurons, if they do anything to the startle, they increase it, because when we inhibit the cholinergic neuron, our startle is less. So that means PPT cholinergic neurons do not impact prepulse inhibition, but they decrease baseline startle. So again, long-standing hypothesis. It's very hard to prove long-standing hypothesis wrong, but this pretty much put the nail into the coffin for that. And now the question is, of course, what other neurons are responsible, and this are my last two slides, I think, is we now look at the glutamatergic neurons. We use a different construct. This, oh no, sorry. First of all, we use a general construct because we thought maybe DREADDs are just not effective enough to inhibit prepulse inhibition. And so we use a construct now that, that is expressed in all PPT neurons, GABAergic, glutamatergic, and cholinergic. And long story short, so we inject, we let it express, we do the designer drug. Now we can inhibit prepulse inhibition when we inhibit all types of pedunculopontine tegmental neurons. So pretty much in line with the lesion experiments. The effect is relatively small because this construct was expressed only in 60 to 70% of the neurons. We also have an effect on baseline startle. So we can recapitulate the lesion studies. So some neurons in the PPT are responsible for prepulse inhibition, but it's not the cholinergic. And so the last construct we use is now a construct that is specific for excitatory neurons because it has a promoter, CaMPARI, which is only active in excitatory neurons. And this way we can target the glutamatergic neurons in the PPT. And there we see the same inhibition of startle response through inhibiting glutamatergic neurons, and an even stronger increase in startle baseline when we inhibit these glutamatergic neurons, which is well in line with former studies of us 10 years ago, where we actually show that they are inhibitory glutamatergic metabotropic receptors on these startle neurons that inhibit startle. We still don't know about the GABAergic neurons, and unfortunately there's no construct that we can use on the GABAergic neurons. So again, maybe a little interim summary. So DREADD inhibition of glutamatergic, but not cholinergic neurons in the PPT impairs PPI and increases baseline startle. We don't know about the GABAergic neurons, and we don't have a construct to actually ask it. And my really last slide now, I show you very preliminary approach that we are trying currently is to actually label the neurons in the PPT that get activated during prepulse inhibition by using a construct which is called CaMPARI, which is a viral construct that contains a protein that is bright green fluorescent and that gets photoconverted by light into a bright red protein, but it also needs high calcium. So what we do is we infuse this, we inject this construct again into the PPT, and we want to label the neurons that are active during prepulse inhibition and therefore have increased calcium, but that are also only active during prepulse inhibition, so only active by sound and in the PPT. So we implant the light fiber into the PPT and provide the light only during prepulse inhibition measurements. And this way we hope that we can label the type of neuron that are specifically active during prepulse inhibition. And then of course, as opposed to in this study, which is a study where they used fiber optometry, we actually then perfuse the animals, get the brains out, and look at which neurons have been photoconverted into red, so which neurons were active during a startle experiment, which neurons responded to the sound, and then do a double labeling and see whether these are GABAergic neurons, cholinergic neurons, or glutamatergic neurons. And with this, I want to end. So we hope that we can at least get a hint of whether GABAergic neurons are involved as well. And then, of course, once we know which neuron type that is, we can specifically test for these neurons in the Cntnap rats. So then we can go back to do immunohistochemistry and electrophysiology and look how neurotransmission of these neurons is affected in the Cntnap rats, which then might account for the disruption in prepulse inhibition. So if you took nothing else from this talk, I hope you took away that sensory processing disruption in autism offers a unique opportunity for translational studies because it's so well conserved between animals and humans. The importance and the difficulty of genetic and environmental animal models. And I want to maybe add one sentence. Our catnap animal has a relatively mild phenotype. And we see the phenotype that I have reported only in the homozygous knockout animals. However, the children in this Amish population that have a fully non-functional catnap protein mutation, they are heterozygous. So there are, although there's this high phase and construct validity of this catnap rat, there are still difficulties or differences to the human situation. And what we're trying at the moment is that we combine a catnap knockout with an environmental factor, with poly-IC, for instance, and to see how these two impacts then interact to create a more severe model of autism. And then I showed you the potential of some innovative novel genetic tools to actually probe for these mechanisms underlying the changes in sensory filtering and sensory processing. And with this, I want to end, and I want to, of course, mention collaborators here at Western and in the Netherlands on this project. And I want to mention trainees. A lot of data I showed was from Kayla Scott, and oops, I forgot Naveen Fulcher. She was mentioned several times here during the talk. Two newer students, Rashka Mabred and Renee, and then my former PhD student, Erin, two postdocs, and my lab manager, Cleusa. And of course, I have to acknowledge all the different funding agencies. And with this, I'll end. Thank you. Shall I stop sharing or shall I?
Yeah, why don't you stop sharing so we can see everybody, and then you can always share again if we need the slides.
Yep.
So that was a great talk. Do we have, do I maybe take like a few questions before we move to the Q&A session? Does anyone have any questions? If not, I have a few questions. So
go ahead.
I was really impressed by really the ability to probe these really specific brain regions and circuits. And it was also interesting to see that some of these changes, longitudinal changes in development were really replicated in the rat model. And I was thinking about this delay in maturation. So does that imply that maybe there is some type of training that could be done to improve some of the auditory brainstem responses, and have people thought about that and what that training would look like for autism spectrum disorder?
Very good question. Yeah, and it's very obvious that while the the brainstem only matures slower, but eventually catches up, that these behavioral readouts persist. And so what we think is, and there's some experimental evidence now as well, what we think happens is that because the brainstem is kind of like lower and slower during development, that there is a compensation at the sensory motor interface in terms of startle, that, you know, that synaptic gain is ramped up because the signals are so low and slow, that then persists. So that, and that makes that behavioral phenotype even worse in the adult animal when, you know, the brainstem then becomes normal, but this increased synaptic gain is still there. And we know that this happens also in hearing when people age and lose hearing abilities, that the synaptic gain is increased in people with hearing deficits, which can become very disturbing. Most of the people, I can't see the people attending this talk, but I assume most of them are not as old, but as you age beyond 50 or 60, a lot of people report that they have this, that background noise and a lot of things become much, much louder. This is, and then they have a hard time to hear things in loud background noise. And so this increased synaptic gain due to a loss of synaptic input is one mechanism that that causes this. And so this is something we, of course, also can test in animals. If this holds true, then it means that if we can increase either the neuronal signals, or the speed of neuronal maturation in the brainstem, we can maybe avoid that. And so there's a lot of effort to actually expose children then, and it's also done in rats, of course, and in mice, to expose these individuals actually to sensory stimuli rather than you know, take the sensory stimuli away, which we often do now with kids with autism. We actually keep them in a quiet and so on. But there's some evidence that this might actually be the wrong thing, that exposing them early on to these sensory stimuli might actually be beneficial. So again, there's a very much translational approach that can directly be tested in the rat, but that we can also test in children with autism. So there's a multimodal aspect to that as well. I only talked about auditory here. But there's also the binding between auditory and visual stimuli are impacted, and we can probe that by using visual prepulses at prepulse inhibition. And so these binding of different modalities of sensory stimuli that also seem to be able to be trained. So if there's a lot of exposure early on, there's evidence that it actually might become better and that these compensations don't occur, and then the behavioral phenotype are not as severe. Good question.
Hi, that was a very nice talk. I just had a quick question about, so you talked about like the glutamatergic neurons being important in the gating. So in your catnap animals, do they have normal numbers of glutamatergic neurons? Do you think it's a loss of neurons that's causing the abnormal gating? Is it the fact that the neurons have abnormal connections, or what do you think is abnormal in these neurons, or are they not there?
So we don't know yet. That's the honest answer. These are things that are underway. So we look in these circuits, in the PPT, in the PNC, whether there are differences. The problem is, of course, there could be differences in a number of neurons that would be easily to quantify, but it might not be a difference in the number of neurons. It might be a difference in the number of synapses, and this is not so easy to quantify. So yes, we are doing patch clamp recordings where we have indirect ways to estimate the number of synapses. Patch clamp recordings in the brainstem are very challenging. So we are at the beginning of that. We did some patch clamp recordings in the cortex, And this is what we saw. In the cortex, in the auditory cortex, we even saw that there is a lower number of inhibitory neurons in the cortex. There, it's the GABAergic neurons that are lower in numbers. Again, in line with that imbalance of excitation inhibition. Again, so we do see that in the Cntnap rats. We really don't know about the brainstem yet. It's unfortunately very hard to do these kind of experiments in the brainstem, since a lot of antibodies that we use in immunohistochemistry do not work in the brainstem. So we use in situ hybridization at the moment. But it's, yeah, so I can't really tell yet. Ask me in 2 years. I hope we have figured it out by then.
Thank you. I just have one other question. Sorry, Megan. So they've looked a lot at, this is a totally different question. They've looked at brain morphology and they see the brain enlarged in various parts of the cerebellum, or there's differences. Some of them are bigger, some of them are smaller. Do you see any differences in the brainstem when they look, or is that is there not enough, too much variability to pick it out?
It's not too much variability, but it's just these nuclei are relatively small. So it's not as easy, and they're not as well organized as the cortex or the cerebellum, where we have these clear layers and so on. So this makes it all a little bit harder. Plus, again, the staining methods are not as reliable in the brainstem. We looked at the Cntnap rat. So again, we looked in the cortex as well. We do see the reported changes in the cortex, the same changes that have been reported for the mice and for some other models. We are looking in the cerebellum. We haven't done it. It's not finished yet. This is underway. I'm not sure if we see, I don't know. And again, in the brainstem, no results yet. So yeah, no.
I was just more curious about in humans, if they had seen anything.
Oh, in humans. In humans, no one looks at the brainstem. They all look at the cortex. So there is, I don't know, there seems to be that, maybe in the cerebellum and maybe thalamus, but there's always this disconnection. This is a joke now, but I have to really say that. But there are these people that are very much focused on the cortex, and they think the brainstem is just there to prop up the cortex. And then the brainstem people always say, oh no, that's the other way around. The cortex only is there to keep the brainstem warm and nice. Of course, it's none of the two, but the brainstem in human research has not been looked at a lot. You can't image that with MRI, or it's very hard to image brainstem because it's such a deep structure. And in the post-mortem studies, there are rarely any studies that look at brainstem.
Thank you.
I have a question, Dr. Schmid. This is a fabulous talk. It gave me many ideas, many knowledge about autism. I'm interested in the sensory filtering, and it's apparently that not in autism, but also in other psychiatric disorders, the sensory filtering is dysfunction. And I'm thinking about breathing or rhythmic respirations effect on the sensory filtering. And it may, maybe it's a literature, go to the literature to give some literature review or some short phrases. It's about, because it will give some direction about how meditation might affect psychiatric disorders or affect autism, help them with the treatment through the breathing method. If you can comment on the breathing, on the sensory filtering in your system, might be very interesting to me.
Okay, breathing and sensory filtering. So this is something I've never thought about, but I'm trying to think quickly. There are two things that come into mind in terms of breathing and sensory filtering. First of all, I think there is, of course, a connection to breathing in Rett syndromes. So these children do not breathe normally. So the breathing circuits are actually affected in at least this kind of, this type of autism, which does not explain why breathing would influence the symptoms, but it's an interesting aspect that breathing circuits can be affected in the same way as other circuits. And so maybe, again, like behavioral therapy, breathing self-therapy could have a beneficial effect. The other thing is, of course, I think through meditation and breathing, again, I have no clue. I've never read about that or really thought about that. But what I could imagine is that the level of arousal, sympathetic versus parasympathetic tone can change. And startle, so a lot of our readouts are greatly influenced by arousal. And so are sensory processing pathways and sensory perception. Arousal has a huge influence on what and how we perceive things. And so it has a huge influence on sensory pathways. And I would be very surprised if breathing and meditation and changes in arousal through that would not impact sensory processing and sensory filtering.
Thank you.